गो लाइव पे, हाँ चलिए ठीक है, एक बार आप देखिएगा। यू आर लाइव, आ गया हूँ मैं, हाँ चलिए ठीक है, ओके 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 बाय। चल yes, yeah, good morning everyone, good morning everyone. I hope yeah, Sakib sir is also here. Good morning everyone. Yeah, yeah, good morning Kapil. Yeah, good morning. I hope audio and visuals are clear to each and everyone there. Audio and visuals are clear to each and everyone there. Yeah, just give me a thumbs up guys that you all are participating in the class right now. And please, yeah, ask your friends to join ASAP. I'll start within like another 2 minutes. Another 2 minutes I'll start the class. So kindly, yeah, Amjad, good morning. Danish, good morning guys. Yeah, Tanu, very good. Sakib, okay. Lakshi, very good. Laris, very good. Legend, Ligand, okay. Anyways, unique names, yeah, everything is clear. Junaid, very good guys. I hope everyone is participating as of now. So yeah, I just, yeah, very good guys. I hope everyone is present over here right now. Yeah, Sanvi. So today we'll start with the basics of molecular biology, in which I will be first discussing the chemistry of nucleotides. That's the toughest topic I suppose, and I will make it a piece of cake for you if you are throughout with me, okay? Like during the entire lecture, if you guys are with me, I will try to make it a piece of cake for you. Yes. So we shall start. I hope like everyone has joined us per now. I mean, yeah, like one more minute and then I'll start with the class, guys. One more minute and then I'll start. Ram, Jangir, Akash, yeah, Raza, very good, guys. Yes. Sure. Okay. जो भी हैं, we'll start with the class now. Yeah. Yeah. We'll start. Arman, very welcome to the class, Arman. Good morning, Arman. Yeah. So let's start. Omkar, very good. I'll just put a text right now. Yeah. Yeah. Kasim, good. Okay, now we'll start. Yeah. Okay. Kamlesh, okay. We'll start with the chemistry of the nucleic acid, guys. So put the heading as the chemistry or the biochemistry of nucleic acids. Chemistry of nucleic acids. Nucleic acids. Yeah, don't worry guys, I will try to cover my maximum portions from anatomy and biochemistry. Yeah, the molecular biology. So don't worry guys, we'll just start. Okay, just start with the class right now. Okay, so just concentrate on what we are doing right now. Apart from that, in the last we'll discuss how to plan up. Okay. So we are now going to start with the chemistry of the nucleic acids. You all know that inside the cell, basically we have the nucleus, and inside the nucleus we have some component that is called as the nucleic acid. Nitin, good morning. Good morning. Yeah. Kritika, good morning. Pranjal, good morning. Yeah. So yeah, so we'll start with the chemistry of the nucleic acids, guys. Yeah. So let's start. So I hope you all know that there are two types of nucleic acids. There are two types of nucleic acids. There are two types of nucleic acid, guys. And remember, these two types are, you already know that according to your +2 knowledge, it is the DNA and the RNA. It is the DNA and the RNA. So remember, guys, that the nucleic acid, it is basically made up of nucleotides. It is made up of nucleotides. You all know that the DNA, the RNAs, okay, these are all made up of nucleotides. Now what is the structure of the nucleotide? We will try to discuss this in detail, guys. We will try to discuss this in detail, guys. And remember, the nucleotide has three components. It has three components, and everyone knows that it is the nitrogenous base, the nitrogenous base, the pentose sugar, and we have the phosphate. We have the phosphate group. We have the phosphate group. So as per now, we have started with the lecture dealing with the basics of the molecular biology, and you all know that all cells contain genes, and these genes are basically responsible for your different fingerprints. Okay, why same members, like different members of the family, okay, they actually appear different, okay, their fingerprints are different. That is all just a result of these genes. Okay, apart from that, guys, you should know that these cells basically contain the nucleic acids, and there are two types of nucleic acid. You already know that it is the DNA, that is the deoxyribose nucleic acid. It is the deoxyribose nucleic acid, and we have the RNA, that is the ribose nucleic acid. We have the RNA, that is the ribose nucleic acid, okay, or ribonucleic acid, deoxyribose nucleic acid or ribose nucleic acid. So we will now study the structures of DNA, RNA. Firstly, we'll try to discuss about the nucleotides now. Yeah. So yeah, many people have joined as of now. I can see in the chat box. I hope everyone has written whatever I have just noted on the screen. Whatever I have just noted on the screen, everyone has written. Yeah. So I'm just waiting everyone to kindly give me a thumbs up. Ki, sir, we are all are with you and we have started with the lecture. Whatever you have written up till now, yeah, we have already mentioned this in our notes. Yeah. So yeah, Rohitash, very good. Junaid, yes. Sanvi, very good. Guys, be very quick in replying to my questions. Okay, whether it is your participation or the questions which I put up on the screen. So Kapil, Manish, very good, guys. I mean, I'm quite impressed that all of you are participating today. And today I will make the nucleic acids piece of cake for you. If you promise me that you will be throughout, like be with me in the lecture, it will be a piece of cake for you guys. Okay. Okay, guys. Perfect, guys. Yes. Everyone, Tanu, Abbas, Shadab, Lakshi, Lokendra, Anshay, Ashwani. Okay, so everyone. Now, guys, firstly, you should know that the nitrogenous base and the pentose sugar, whenever these both are combined, they are this structure. It is called as nucleoside. This structure it is called as nucleoside. Okay, this structure it is called as nucleoside. So now first what we will do, we will try to study each of these structures in detail. We'll try to study each of these structures in detail, guys. So starting with the nitrogenous base, guys. Starting with the nitrogenous base. So you all know that we have two types of nitrogenous base. We have two types of nitrogenous base, and you all are aware of this, that these are purines. These are purines, and we have pyrimidines also. We have pyrimidines also. So we have the purines, we have the pyrimidines also. So now guys, remember, we will first study the structure of the purine. We will first try to study the structure of the purine. So remember that purines are basically composed of two rings. There are two rings. So if suppose I'm drawing a purine right now in front of you. So one ring is basically a 6-membered ring. Okay, it is a 6-membered ring, and the other ring is basically a 5-membered ring. Okay, the other ring is basically a 5-membered ring. So we have one ring, we have the second ring. Just mention it right now. We have two rings. Yeah, everyone is participating. I'm quite happy about that. Yeah, be with me guys, you will all master today in this. Yeah, Vyomkesh, welcome to the class. Yeah, so we have two rings, guys. Okay, now these are heterogeneous rings. These are heterogeneous rings. Okay, these are different rings. You can very well appreciate in the on the screen, guys. You can very well appreciate that these are different rings. Okay, one is of 6 carbons, one is of 5 carbons. Yeah, now how many purines are there? First try to remember that. Okay, just first let us understand the structure, guys. Let us understand the structure. Now remember that the numbering of these rings, how it is done. First try to understand. Now suppose this is the ring. Okay, this is the ring. How the numbering is done, guys, try to remember. The numbering is started from this particular point. The numbering is basically started from this particular point. So it will be 1. Okay, it will be 2, it will be 3, it will be 4, it will be 5, it will be 6. Okay, and see, the numbering basically started at this particular point, and it is actually done in an anti-clockwise direction. It is actually done in an anti-clockwise direction. Yeah, it is actually done in an anti-clockwise direction in the first ring. Coming on to the second ring, guys, it is done in a clockwise direction. Till 6 you have done. Now it is the 7, 8 and 9. It is 7, 8 and 9. Okay, so over here it is the clockwise direction. Over here it is the clockwise direction. So you just see that we have started with the first, okay, from this particular point, completing this particular ring in an anti-clockwise direction and moving on to the other ring in a clockwise direction. This is how the numbering of the carbon atoms is done. This is how the numbering of the carbon atoms is done. Yeah, now guys, try to understand that I am telling that all odd positions, that is just a trick, guys. It is a trick. All odd positions will occupy nitrogen except 5th place. Okay, except the 5th one, or at 5th place there is no nitrogen. So starting with 1 is an odd number, guys. So yes, over here the nitrogen will be there. So I'm just drawing a structure over here. So that is the first position. You can see I'm saying that all the odd positions will occupy nitrogen. Coming on to the second is an even, no need of odd position. Coming on to the third, yes, third is an odd number, it will have nitrogen. Apart from that, going on to the fourth के पास नहीं होगा. Coming on to the fifth is an odd number, but I told you it's an exception. Over here there will be no nitrogen. Sixth is an even number, no nitrogen. Coming on to the other cyclic ring, the seventh position, yes guys, seventh is an odd number, yes, nitrogen will be there. Coming on to the eighth, no, it's an even number. Coming on to ninth basically it is an odd number. Over here it will be the nitrogen will be present. So this is how the numbering is done. Okay, this is how the numbering is done. Okay, this is how the numbering is done. And remember, it is started from this particular point. Okay, and all the odd positions will be actually occupied by the nitrogen. Balancing is not important in biochemistry. It is generally the simple architecture that you guys should know. It is generally the simple architecture that you guys should know. Okay, so remember that if you just want to balance, you know that nitrogen basically has to occupy 3 valency. Okay, so ultimately there will be a double bond over here, a double bond here, a double bond here, a double bond here. Okay, so in that case, all the 3 valency of the nitrogen, you can very well appreciate from the picture as well, the 3 valency of the nitrogen, it is occupied. Okay, so this is the general structure of the purine. It is the general structure of the purine. General structure of the purine. We are not talking about phosphodiester bond, that is in the last of this lecture. We are just studying the basic structure guys now. It is the general structure of purine. So yeah, so guys, let me know that if you all understood the basic structure of the purine. Let me know guys if you all understood the basic structure of the purine. Yeah, I'm waiting for your answers guys. Just give me a thumbs up that you all understood the basic structure of the purine. Yeah guys, if you will be with me, yeah, Nahid, Sanvi, Junaid, Qasim, Ronak, very good guys. Laris, Kumud, Danish, Lakshi, I'm quite happy that you all guys are participating as of now. Yeah, I'm very happy that you guys are participating. Uzma, Vyomkesh, very good guys. So yeah, yes, Amjad, very good. Anshay, Manish, Abbas, Lokendra, Uma, exceptions are always there in biology, and so by your name, whosoever he is or she is. Anyways, Sagar, Vyomkesh, very good, Pavan, very good guys. I mean, I'm quite happy that you all are participating. So that is a general structure of the purine. Now remember how to remember, there are 2 purines guys. So first we are just discussing about the purine, and you should know that basically we have 2 purines, and remember the names are adenine and guanine. Adenine and guanine. So remember, how to remember sir, हम तो बार बार भूल जाते हैं. So remember by the help of a mnemonic that all girls are pure. So yeah, all stands for adenine. G stands for guanine, and P stands here for purines. So remember that adenine and guanine are purines. Adenine and guanine are purines. Welcome Jay, welcome to the class. Yeah. So yeah, adenine and guanine are purines, and remember, apart from that guys, you have to also remember about the minor purines guys. You have to also remember the minor purines. Now we have 3 minor purines, that is xanthine, hypoxanthine, and along with that we have the uric acid. The uric acid. Okay. So these are the minor purines. Now guys, we will first learn the structures of adenine and guanine. We'll first learn the structures of adenine and guanine. Now, Now guys, you know the basic structure of a purine. You know the basic structure of a purine. It is basically a heterocyclic ring occupying nitrogens at the odd positions. Occupying nitrogens at the odd positions. Okay. And you can very well appreciate that, we are starting the numbering from here: 1, 2, 3, 4, 5, 6, 7, 8 and 9. Balancing is not important over here right now. I'm just concentrating on the general architecture now. General architecture. So you all can understand ki, sir, the odd positions are basically occupied by the nitrogen atoms except the fifth one. Apart from that guys, this is the general structure of the purine. It is a purine. Okay, it is a purine composed of two heterocyclic rings. Okay, heterogeneous rings are there. Okay, one is a 6-membered ring, the other one is a 5-membered ring. Now guys, what is the structure of the adenine? Coming on to the structure of the adenine guys, see, the structure remains the same. The structure remains the same. Okay, so I'll just draw it. Yeah, the structure remains the same, but what extra you will find over here? But what extra you guys will find over here? Okay, so yeah, you will have a amine group over here. You will have a amine group over here. Sayed, Saqib, welcome guys. You have a amine group over here. Dikshita is also here. Yeah, Manish. Okay guys. So you have an amine group over here. That is the simple, like you have to know the structure of the adenine. Can anyone tell me the name of this particular structure, like by formula, like giving the numbers 1, 2, 3, 4, 5, 6, 7, 8 and 9. Now can anyone tell me कि 6th position पे मैंने कौन सा group लगाया? Which group is this? I'm waiting for your answers guys. Which group is this that I have put on the screen? Yeah, I'm waiting for your answers guys. Which is the group that I have put on the screen? Amine group. Very good, Junaid. I mean, I expected that you will give the answer. What about rest, guys? Are you all satisfied with the answer given by Junaid? Yes. Poonam, Ronak, Akash, Laris, Kasim, Dev. Very good. Very good, guys. Yes, Bhupender. Yeah, you are very, very right about the answer as well. Amjad, Rasab, Tanish, Chaman, Nahid, Lakshi. Very good. I mean, so many students are participating today. So, guys, remember the fact. Yeah. इट इज एट 6th पोजीशन. तो एडेनिन का नाम तो आप लोगों को खुद ही पता चल गया कि सर ये 6th पोजीशन पे है और ये अमीन ग्रुप है. ये अमीन ग्रुप है. NH2 को अमीन ग्रुप बोलते हैं. तो ये क्या हो जाएगा? 6 अमीनो प्यूरीन. 6 अमीनो प्यूरीन इज द अदर नेम ऑफ एडेनिन. इट इज द अदर नेम ऑफ एडेनिन गाइस. इट इज द अदर नेम ऑफ एडेनिन. ओके. So everyone is perfect with the naming of adenine. Now coming on to the structure of guanine. कि सर सब में तो सेम ही है. प्यूरिन तो एक ही जगह रहेगा, तो चाहे ये वाला स्ट्रक्चर हो चाहे गुआनिन वाला स्ट्रक्चर हो. क्या एक्स्ट्रा है उसके हिसाब से नेमिंग करेंगे. विच स्ट्रक्चर इज एक्स्ट्रा उसके हिसाब से नेमिंग होगी. चलो अब गुआनिन का स्ट्रक्चर बनाते हैं और फटाफट से इसकी नेमिंग करते हैं. उसके बाद सारे बॉन्ड्स बताऊंगा. मतलब आज की क्लास अगर आप लोग अटेंड कर लोगे तो यू विल एक्चुअली लर्न अ लॉट. Okay, yeah, so yeah, just starting with the same pattern. ठीक है, just starting with the same pattern. And remember guys, yeah, remember that following my same rule कि all odd positions will be occupied by the nitrogen except the fifth one. Okay, apart from that guanine में ऐसा क्या एक्स्ट्रा है सर? guanine में second position पर एक amine group है NH2, that is the amine group at second position, and at sixth position there is an oxy group. At sixth position there is an oxy group. Now students, can anyone tell me the name of the guanine, the other name of the guanine, just as we talked about adenine? Yeah, so I'm waiting for your answers. Yeah. Yes, anyone would like to share what will be the name? No one, no one. Okay. No. Just other name of Amjad, other name of the like this particular structure. See, guanine can be written as Rasab Bhupender. Okay. Guys, I just wanted the name of this particular structure. You can very well appreciate कि सर सेकेंड पोजीशन पे अमीन है तो इट विल बी 2 अमीनो 6 पोजीशन पे ऑक्सीजन ग्रुप है तो इट विल बी 6 ऑक्सो प्यूरीन. इट विल बी 6 ऑक्सो प्यूरीन. सो दैट इज द अदर नेम फॉर गुआनिन कि सर सेकेंड पोजीशन पर अमीन ग्रुप है, 6 पोजीशन पर ऑक्सो ग्रुप है तो द नेम विल बी 2 amino 6 oxo purine. We are just discussing about the bases now. You all know that the nucleotides are basically made up of base, nitrogenous base, that is the purines and pyrimidines, pentose sugar and the phosphate group. Yeah, we'll just write the simplest name over here, guys. That is 2 amino 6 oxo purine. 2 amino 6 oxo purine. Now starting up with the next part, that is the pyrimidine. So guys, just give me a thumbs up that you all have understood the structures of the pyrimidines, purines. You all understood the basic structure of the purines. Just give me a thumbs up, guys. So we'll start with pyrimidines now. Okay, we will start with pyrimidines now. Yes. Yeah, Laris, very good. Laris, very good. Junaid, Hansi, very good. Vijay, Nahid, Usma, very good. Guys, whosoever is participating, like on daily basis, if you are listening the lectures and even doing your like 2-hour study, self-study, that is enough for qualifying the exam. So classes are must. Yeah, Soumya, very good, guys. I mean, I'm totally impressed if you guys are understanding the मतलब कितने difficult structures लगते हैं. I have just made it a like piece of cake for you guys. Yes, exception. Yeah, Sagar, very good, guys. Very good. Okay. Danish, Kapil, very good, guys. So we'll now focus on the structures of pyrimidines now. So you all should know that pyrimidines are basically a single heterogeneous ring, and remember, सर आप कमाल हो. जुनैद, ये कमेंट मैंने तुम्ही से एक्सपेक्ट किया था. I mean, anyways, so yeah, so they have a single heterogeneous ring. अब सर इसका स्ट्रक्चर कैसे बनाएंगे? वही 6 कार्बन वाला एक साइकिल बना लो, hexagonal structure like this. Now guys, try to number it from the position where I am showing you right now on the screen. Yeah, try to number it from the position, guys, where I am showing you right now on the screen. This is the first carbon. Number in the pyrimidine is starting, it started from the below, okay? It's the first carbon, okay? The second one, okay? The third one, the fourth one, guys, the fifth one, and the sixth one. So it is a single height, it is a single like heterogeneous ring, okay? Numbering is starting from the first carbon going up till like this in an anti-clockwise manner. You can very well appreciate. Yeah, it is going in an anti-clockwise manner, guys. Now, guys, the same rule is applied. The same rule that I have asked you, that all places, all odd places will occupy nitrogen, except fifth place. So I'm just transforming the structure to the one statement which I have mentioned on the screen. Now again, drawing. So the first is basically this particular structure. So the first carbon, that is an odd number. Yeah, Nitin Kaushik, yeah, from Bishkek. Very good, guys. Yeah, very good. I'm quite happy that you all are participating as of now. Yeah, so just the first carbon, it is an odd number. हाँ सर, यहाँ पे नाइट्रोजन आएगा. अकॉर्डिंग टू रूल आपने तो यही बोला है कि यहाँ पे नाइट्रोजन आएगा. सेकेंड प्लेस इवन है सर, यहाँ नहीं आएगा. थर्ड प्लेस पर आ जाओ. थर्ड इज एन ऑड नंबर. हाँ सर, यहाँ आएगा. फोर्थ इवन है. फिफ्थ पे सर आपने एक्सेप्शन दिया है, दो इट इज एन ऑड नंबर, यहाँ पर नाइट्रोजन नहीं आएगा. कमिंग ऑन टू द सिक्स्थ इज एन इवन नंबर. So that is the basic structure of the pyrimidine. That is the basic structure of the pyrimidine. नाइट्रोजन के 3 वैलेंसी पूरी करनी होती है और कार्बन की 4 होनी चाहिए. ठीक है. So yeah. नहीं, यहाँ पे जरूरत नहीं है. Wait. Yeah. नाइट्रोजन की 3 वैलेंसी पूरी होनी चाहिए. Okay, so yeah, that is the basic structure. Balancing is not important in biochemistry. I've just told you, just the basic structure you should know. Yeah, now guys, try to appreciate the fact that we have different types of pyrimidines. Now sir, how to learn the different types of pyrimidine? आपने purines का तो बता दिया. अब now how to learn the different types of pyrimidines will again have a very simple trick. You know that pyrimidines contain a letter Y. So we have 2 types of pyrimidines, guys. That 3 types basically. Okay, one is like okay, these are the thymine, cytosine, and uracil. Thymine, cytosine, and uracil. It is like Y. Y is coming, okay, uracil में नहीं है. Or you can otherwise learn it from a mnemonic that pyrimidines are cut from purines. Pyrimidines are cut from purines. देखो purines में 2 रिंग्स थी ना, और प्यूरिन की एक रिंग को अगर हम हटा दें तो वी विल गेट अ pyrimidine. तो दैट इज द बेसिक स्ट्रक्चर. और ये जो कट मैंने आप लोगों को लिखवाया है, दैट इज CUT. तो ये कट आपका निमोनिक ही है, दैट इज द साइटोसिन, यूरासिल एंड थाइमीन. दैट इज द साइटोसिन, यूरासिल एंड थाइमीन. So hence by the mnemonic also, by just Y letter also you can remember, or you just know the simple architecture कि सर हाँ, pyrimidines are cut from purine. Purine heterocyclic ring होती है, ठीक है. उसमें से अगर दूसरी वाली साइकिल जो है रिंग, वो अगर हम रिमूव कर देंगे तो we'll get a pyrimidine. So pyrimidines are structures which are cut from purine, and cut is nothing but the cytosine, uracil and thymine. The cytosine, uracil and thymine. Yeah, yes guys. So yes, I hope everyone is over here. Like we are reaching quite number of students over here. Yeah, don't skip the class guys. I will not make this video available later on. I'm pretty sure about this. Ask your friends to join the live session ASAP. I will not make the video available later on. Yeah, so it's very necessary to attend the live session at that particular point. Notes तो कहीं से भी मिल जाएंगे, but thing is the explanation part is required. I'm just discussing the structures which are mentioned in the book. Just yeah, everyone, Lakshi, Kumud, Laris, Sanvi, Anshay, Manish, Pavan, Kasim, very good guys. I'm quite impressed that you all are along with me. Yeah. Now we will discuss the different structures of pyrimidines. Kapil, very good. Now we will discuss the different pyrimidines. Okay, we'll write the heading as structures of different pyrimidines. Okay, structures of different pyrimidines. Very good. Nahid, very good. Yeah, Amjad, Abhishek, जुनैद अलग है तभी तो चल रहा है ना मार्केट में, एक जैसा होता तो नहीं चलता. Razab, Shadab, Abbas, very good. Shakir, very good. I'm quite impressed that everyone is over here. Yeah. Structures of different pyrimidines. Structures of different pyrimidines guys. अब सर बेसिक स्ट्रक्चर तो आपने हमें समझा दिया कि कोई भी pyrimidine हो, चाहे यूरासिल हो, थाइमीन हो, उस सबका स्ट्रक्चर तो एक जैसा ही होगा, बट उसमें क्या एडिशनल होगा सर, ये आप हमें बताओगे. सबका स्ट्रक्चर यहां से स्टार्ट होगा 1 2 3 4 5 6, इस पॉइंट से नंबरिंग स्टार्ट होगी सर. और तीनों स्ट्रक्चर्स साइटोसिन, यूरासिल, थाइमीन, सबके स्ट्रक्चर्स विल एक्चुअली रिमेन द सेम and the numbering will remain the same. The nitrogens will occupy the odd positions except the fifth one. So yes, so all the three structures are absolutely the same. कि सर ये सारे तो pyrimidines ही हैं, ये भी pyrimidine है, ये भी pyrimidine है, and this is also a pyrimidine. Now starting with the structure of the uracil guys. सेकेंड पोजीशन पर यूरासिल में एक ऑक्सीजन ग्रुप लगा होता है. ओके, और एक फोर्थ पोजीशन पर ऑक्सीजन ग्रुप लगा होगा. एंड नाउ गाइस, कैन एनीवन टेल मी द नेम ऑफ दिस पर्टिकुलर यूरासिल? सर केमिकल नेम बताइए फटाफट इन द चैट बॉक्स राइट नाउ गाइस, इन द चैट बॉक्स. I'm waiting for your answers guys. Yeah, I'm waiting for your answers. टू फोर ऑक्सीपिरिमिडिन 2,4 डायोन. ठीक है, थोड़ा कॉम्प्लेक्स, इजी बनाएंगे इसको थोड़ा. यार 1 और 3 पे तो है ही नहीं ना, 2 और 4 पे है. रसब 2 और 4 पर है बच्चे. 2,4 डाई ऑक्सीपिरिमिडिन. सानवी, कासिम, भूपेंद्र, जुनैद, वेरी गुड. हाँ लारिस, इसको थोड़ा सा इजी बनाएंगे हम लोग. देखो, सेकेंड पोजीशन पे ऑक्सीजन ग्रुप है, फोर्थ पोजीशन पर ऑक्सीजन ग्रुप है, तो दोनों ग्रुप सेम है, तो 2 और 4 मैं साथ लिखूंगा. डाईऑक्सी, डाई मतलब दो जगह पे हो गया, या डाईऑक्सी, ओके, डाईऑक्सी pyrimidine. So that is the other name of the uracil, guys. That is the other name of the uracil. And remember that uracil is found in RNA only. Uracil is found in RNA only. Yeah, everyone is right. That's why, guys, I just put you questions so that interaction should be there. कि सेकेंड और फोर्थ पोजीशन पे सर एक ही ग्रुप लगा हुआ है, दैट इज द ऑक्सीजन ग्रुप, और वो ऑक्सीजन ग्रुप लाइक द नेम बाय द नंबरिंग कि सर सेकेंड पोजीशन पे आया, फोर्थ पोजीशन पे आया, और दोनों जगह एक ही ग्रुप है, तो 2 and 4 विल बी क्लब टुगेदर, डाईऑक्सी, दो ऑक्सीजन ग्रुप हो गए, और पिरिमिडीन तो पिरिमिडीन ही है, बेसिक स्ट्रक्चर तो वही रहेगा. Now guys, coming on to the next one, that is the thymine, guys. Thymine, and remember that thymine is basically present in DNA. Thymine is basically present in DNA only. And you guys will tell me कि sir second position पे भी oxygen group है, fourth position पर भी oxygen group है. It is just similar to uracil, but in addition to this, it will have a methyl group at the fifth position. It will have a methyl group at the fifth position. जैसा कोई बात नहीं यार, गलती करोगे तभी सीखोगे, ठीक है, गलती होगी तभी सीखोगे, तो इट्स द बेस्ट वे. अभी लर्निंग फेज में हो, सब सीखना है, ठीक है. इवन आई डिडंट न्यू द स्ट्रक्चर्स बिफोर, मतलब तो इट्स जस्ट लाइक कि आप प्रैक्टिस कर रहे हो, समझ रहे हो चीज़ों को, तो धीरे धीरे सब आ जाएगा. और इसी तरीके से भी होनी चाहिए. क्लास अटेंड कर रहे हो, दैट इज अगेन अ पॉजिटिव पॉइंट, ओके. So guys, now tell me the name of this thymine. Thymine तो just uracil की तरह है, 2 पोजीशन पे भी ऑक्सीजन है, फोर्थ पे भी है, बट एडिशन टू दैट फिफ्थ पर मिथाइल ग्रुप होगा, फिफ्थ पर मिथाइल ग्रुप होगा. नाउ कैन एनीवन टेल मी व्हाट विल बी द नेम ऑफ दिस पर्टिकुलर thymine? या वेटिंग फॉर योर आंसर्स गाइस. या 2,4 हाइड्रोक्सी ग्रुप तो है ही नहीं, 5 मिथाइल 2,4 डाईऑक्सी पिरिमिडीन, ना 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 ना, हाँ ओके, हाँ ठीक है, ओके, तुमने उस वे से पैटर्न फॉलो किया है, ठीक है, हाँ वेरी गुड. बट बेटा नंबरिंग पहले हम लोग देते हैं ना, तो ठीक है. तो जस्ट गाइस रिमेंबर दैट इट विल बी 2,4 डाईऑक्सी 5 मिथाइल pyrimidine, 5 methyl pyrimidine. Yeah, simplest way to just write it like that. Dioxy हो गया ना यार, D आ गया ना पहले, है ना, dioxy आ गया. So yes, it will be 2,4 dioxy 5 methyl pyrimidine. सर सेकंड पोजीशन पे भी ऑक्सीजन ग्रुप है, फोर्थ पे भी है, 2,4 डाईऑक्सी हो गया, और फिफ्थ पे CH3, दैट इज द मिथाइल ग्रुप. तो इट विल बी 2,4 डाईऑक्सी 5 मिथाइल pyrimidine. 2,4 डाईऑक्सी 5 मिथाइल, यस साकिर नाहिद, या 5 मिथाइल यूरासिल, ऐसे नहीं बोलेंगे, देखो कुछ रिएक्शंस होती है. चलो आई जस्ट क्लियर इट आफ्टर सम टाइम, जस्ट अनदर कपल ऑफ मिनट्स. पहले हम ये कर लें और फिर मैं बताता हूँ कि आपस में इंटरचेंज कैसे कहते हैं. नाम से नहीं करना है, कुछ रिएक्शंस होती हैं, तो वो अभी हम देख लेंगे. बस थोड़ा सा हिंट दे दूंगा, तो जब हम लोग बाद में पढ़ेंगे और डिटेल में तो पता चलेगा. तो पिरिमिडीन लास्ट वाला सबसे जो इंपॉर्टेंट है, दैट इज द साइटोसिन गाइस, दैट इज द साइटोसिन. तो साइटोसिन इट इज प्रेजेंट इन बोथ आरएनए एंड डीएनए. इट इज प्रेजेंट इन बोथ आरएनए and DNA. It is present in both RNA and DNA. And remember that cytosine में second position पर oxygen group है, और fourth position पर, remember guys, fourth position पर, just I mentioned bond wrong हो गया, yeah, fourth position पर there will be an amine group over here. There will be an amine group over here. Yeah. So at fourth position there will be an amine group. Now can anyone tell me the name of the cytosine, the chemical name? Anyone? Yeah, I'm just looking at the chat box, and just a simple name कि sir pyrimidine तो है ही, सेकेंड प्लेस पे ऑक्सीजन ग्रुप है, एंड ऑन द फोर्थ प्लेस देयर विल बी अ अमीन ग्रुप. सो क्या नाम होगा? कैन एनीवन टेल मी? या, waiting for the answers guys. I hope everyone should participate. Everyone. Yeah, 2-oxy 4-amino pyrimidine. Very good. Bhupinder, Amjad, I'm quite impressed that you all are like perfectly. You all are like perfectly, yeah, answering the questions. Yes, answering the questions. Yeah, it is 2-amino. Oh, sorry, 2-oxy. Yeah, 2-oxo. Okay, 4-amino pyrimidine. Okay, 2-oxo oxygen group second position, and an amine group at the fourth position. 2-oxo 4-amino pyrimidine. It is the basic name. Yes, everyone is right. Sakir, Qasim, Abbas, Nahid, Lakshi, Manish, very good guys. I am quite impressed that all of you are participating. Kumud, very good. Kumud, very good. Very good guys. जो डेली पढ़ रहे हैं, except for all the tantrums, so they will actually get a fruitful result. Okay, consistency is the key to success, remember that. And if you're consistent throughout the studies, throughout your MBBS years, I mean, you will get a fruitful result thereafter. You won't have to spend a lot of energy studying at last, और इतनी टेंशन लेने की ज़रूरत नहीं पड़ेगी अगर सिंपली क्लास में अटेंटिवली सुन रहे हो और नोट्स बना रहे हो. नोट्स बनाइए और समझते जाइए चीज़ों को कंसेप्चुअली. तो बेसिक स्ट्रक्चर्स अब प्यूरिन्स और पिरिमिडीन्स सबको आ गए हैं. सो जस्ट गिव मी अ थम्स अप गाइस. यस, शदाब, अमजद, अंशे, वेरी गुड गाइस. Just give me a thumbs up that you all have understood the basic nitrogenous bases. कि सर बेस तो सारे ही समझ में आ गए. बेस तो सर एडेनिन, गुआनिन, साइटोसिल, यूरासिल, थाइमिन पहले नहीं आते थे, अब अच्छे से आ गए सर. या, वेरी गुड. अंशे, लक्ष्य, नाहिद, विजय, यस यस, हंसी, वेरी गुड गाइस, वेरी गुड, वेरी गुड. यस, Omkar, Junaid, very good, very good. Sakib, Shakir, Kumud, Ronak, Bhupendra. I'm quite impressed. Like many of the students are attending the class today. Yeah, Tanish, Shakir, yes, Dev, Heather, very good. Soumya, very good. Manish, very good. Very good, guys. I'm quite impressed. Yes. Now, guys, we will move to the structure of the nucleoside now. We will start with the structure of the nucleoside. Crystal clear. क्रिस्टल क्लियर होना चाहिए यार. बहुत इजीली तरीके से, बहुत अच्छे ट्रिक्स देके तुम लोगों को मैंने स्ट्रक्चर्स क्लियर की. रटने से बेटर है मेरी ट्रिक याद रखो. एक ही स्ट्रक्चर सब जगह है, बस डिफरेंट डिफरेंट पोजीशंस पे. पोजीशन की नंबरिंग भी सेम जगह है, चाहे प्यूरिन्स हो चाहे पिरिमिडीन्स हो. उनमें क्या क्या ग्रुप्स ऐड हो रहे हैं बस उतना ध्यान रखना. डाटा सॉल्व सेट, मतलब इसके बाद तो लाइक सबसे ईजीएस्ट पार्ट है ये. नाउ यू ऑल नो दैट गाइस दैट द न्यूक्लिक एसिड्स, the nucleic acids are basically the DNA and RNA, and these nucleic acids are basically made up of nucleotides, and you all know that these nucleotides are further made up of, yes, these nucleotides are further made up of nitrogenous base, phosphate and sugar. Yes, and that sugar is basically the pentose sugar. Nitrogenous bases we have already discussed कि सर purines होते हैं and we have pyrimidines. Purines and we have pyrimidines. Purines are basically, all girls are pure, you all know that. Adenine, guanine, okay, all girls are pure, and pyrimidines are basically the cut. Cytosine, uracil, thymine. We have all discussed this. We have already discussed the structures of all these 5 important nitrogenous bases. Now guys, coming on to the pentose sugar. Okay, coming on to the pentose sugar, or more precisely I will write it now. You all know that कि जब nitrogenous base plus pentose sugar आपस में मिलेंगे तो there is a formation of nucleoside. There is formation of the nucleoside. So now nucleoside we will start. Now guys, remember, this is the basic structure of a purine. Now if suppose this is a basic structure of the purine, I told you it is a heterocyclic ring. Okay, it is a heterocyclic ring. Numbers are basically starting from over here: 1, 2, 3, 4, 5, 6, 7, 8 and 9. You all know that all the odd positions will occupy the nitrogen except the fifth one. Now this is basic structure of the purine. Now let us take this particular example. Okay, now this N nitrogen atom of the 9th, this is actually, can anyone tell me meanwhile which purine I have drawn on the board? I want the answers. Meanwhile, I'll just continue over here. And remember that this particular nitrogen is attached to a pentose sugar. Now this is the structure of a pentose sugar. कि सर ये होता है pentose sugar का structure. This is the structure of a pentose sugar, guys. Now guys, remember that the pentose sugar की numbering, pentose क्यों? क्योंकि we have the 5. Yeah, we have guanine, adenine as the answer. Okay, what about else? जुनैद ने guanine बोला है. What about else? Yeah, adenine है ना यार, guanine कैसे हो जाएगा? या जुनैद, या इट इज एडेनिन गाइस, एडेनिन. 6 अमीनो प्यूरिन हो गया ना, 6 अमीनो प्यूरिन हो गया. ओके, 6 अमीनो प्यूरिन हो गया ना, 6 पोजीशन पे अमीन ग्रुप जुड़ा हुआ है तो इट्स 6 अमीनो प्यूरिन. दैट इज द नाइट्रोजनस बेस, एंड इट इज द पेंटोज शुगर. अब हम न्यूक्लियोसाइड बनाने की कोशिश करें. सो जस्ट लिसन कि सर न्यूक्लियोसाइड पेंटोज शुगर का बना हुआ है, मतलब 5 रिंग्स हैं इसके अंदर, ठीक है. और इसकी नंबरिंग यहाँ से शुरू होती है इस पॉइंट से, और इसको हम लोग C1 प्राइम बोलेंगे, C2 प्राइम बोलेंगे, C3 प्राइम बोलेंगे, C4 प्राइम बोलेंगे. और रिमेंबर that this particular structure is attached to another carbon over here that you all should know that it is the CH2OH. Yes, typing mistake. Okay, CH2OH. और ये वाला जो कार्बन है that is the fifth carbon guys, and that is the C5 prime carbon. Now everyone will ask me कि सर हमने यहाँ पे कार्बन के ऊपर ये प्राइम का निशान क्यों बनाया? Why we made like a prime on this particular carbon or all the carbons of the pentose sugar? क्योंकि बच्चे, we all know that in the nitrogenous base we also have carbons. To differentiate it from the carbons of the nitrogenous base, we actually made a prime sign on the carbons of the pentose sugar. ये प्राइम साइन इसलिए लगाते हैं क्योंकि नाइट्रोजनस बेस के अंदर भी तो कार्बन एटम है. उस कार्बन से इस कार्बन को डिफरेंशिएट करने के लिए, जस्ट टू डिफरेंशिएट दैट, हमने प्राइम साइन लगाया. Just to differentiate that, we have put a prime sign, guys. Just to differentiate that, we have prime sign. Yes, Laris, very good, Laris, very good. Now, guys, remember, so that is the basic pentose sugar you should know. Now, I just wanted to, okay, so just write it over here now. Now, suppose this is a pentose sugar. Put the heading as pentose sugar, guys, and everyone can appreciate कि सर pentose ring है और ये तो यहाँ CH2OH, ये 5 कार्बन हो गए, C1 प्राइम, C2 प्राइम, C3 प्राइम, C4 प्राइम, और ये वाला कार्बन जो है वो C5 प्राइम कार्बन है. So we have all these 5 carbons. We know that. Okay. All these 5 carbons are done. और ये बीच में ऑक्सीजन ग्रुप भी होता है. I'm just not drawn it. Yeah, there's an oxygen group in between. Just mention it over here as well. Okay, there's an oxygen group over here. Now, point is not about that. Now, guys, there are 2 types of pentose sugar, ribose and deoxyribose. Okay, so we have 2 types of pentose sugar, ribose sugar and deoxyribose sugar. Deoxyribose sugar. Now, how to differentiate, sir? Ribose और deoxyribose में differentiate कैसे करेंगे? So simple trick. If OH group is present at C2 prime and C3 prime, then it is ribose sugar. If OH group is present at 2 prime or 3 prime position, then it is the ribose sugar. Okay, ribose pentose sugar. Okay, and now suppose if I draw another diagram, and you all know that it is a 5 carbon ring, C prime, C2, C3, okay, C4 and CH2OH, that this carbon is the C5 prime carbon. Now if suppose the OH ring is present only at 3 position, that is C3 prime position, then it is actually the deoxyribose sugar. If OH, yeah, shut up, you're right. I don't know which part you are talking about. Yeah. See, if OH group is present at 3 prime position, then it is deoxyribose sugar. Simple trick, sir. सेकेंड और थर्ड कार्बन पर OH होगा तो राइबोस, और सिर्फ थर्ड पर होगा तो डीऑक्सीराइबोस. That is how you differentiate between a ribose and a deoxyribose sugar. So just give me a thumbs up, guys, that all of you understood as of now. And yeah, all of you understood as of now. Just give me a thumbs up, guys. Nahid, very good, Nahid. Junaid, yes, very good. Razab, very good. Usma, very good. You guys will rock in the next exam. If you are continuously attending my lectures without any fail, guys, you will rock the exam. To all the ones who are attending without fail, without excuses, Jaipur, Laris, Meet, Ronak, Pavan, Amshad, Hansi, Danish, very good. If you will attend it without fail, I'm guaranteeing you guys at once you will qualify the next exam. Yes. So let's continue to draw. Yeah. Let us continue to draw the nucleoside now. Nucleoside formation. And remember that I have taken a simple structure of a purine. Okay. Nucleoside formation, basically it is the nitrogenous base plus pentose sugar. Nitrogenous base plus pentose sugar. and you all know that we have the starting from here, yeah, all the odd positions will occupy the nitrogen except the fifth one. And suppose we are taking purine, and which purine we are taking? We are taking adenine. Now we are taking adenine, okay, we are taking adenine. And remember guys that now adenine is basically a purine and purine is basically a nitrogenous base. So nitrogenous base when combined with pentose sugar, it will form the nucleoside. Nitrogenous base when combined with pentose sugar, it will form the nucleoside. Now guys, I'm trying to draw a pentose sugar with a different color. Okay. Now this is a pentose sugar and you can appreciate that this is the C1, it is the C2, C1 prime. Don't forget the prime, just to differentiate between the carbon atoms of the purines, okay, the nitrogenous bases, the C3, the C4 prime, and ultimately we have CH2OH, that is the C5 prime carbon. Now guys, remember N9 and C1 prime, these two carbon atoms are joined together, okay, these two carbon atoms are joined together, and this bond is basically called as the beta N glycosidic bond, beta N glycosidic bond, and it is the bond between C1 prime of pentose sugar and N9, the nitrogen that is at the ninth position, and the C1 prime, N9 of purine, N9 of purine. Now guys, if I tell you that suppose we take a different structure, okay, we take a different structure, that is a pyrimidine, any pyrimidine, okay, and this is the nitrogen that is at first position, and suppose this is a pentose sugar, okay, that is C1 prime, C2 prime, C3 prime, C4 prime, and CH2OH, that is the C5 prime. We have 5 carbons. Now N1 basically joins with C1 prime in case of pyrimidine. So you all know that this particular structure, it is a pyrimidine. Pyrimidine basically it is a single structure, okay, single heterocyclic ring, okay, and remember that N1 is basically joining with C1 prime of this particular carbon, and that too I will indicate with a bond, okay, that too I will indicate with a different color. Let us suppose say this color, okay. So that is basically the nucleoside formation, and that is again a beta N glycosidic bond, beta N glycosidic bond, okay, beta N glycosidic bond, and remember it is between N1 of pyrimidine and C1 prime of ribose sugar, C1 prime of ribose sugar. I hope the beta N glycosidic bond is clear to all of you. The beta N glycosidic bond is clear to all of you. Just give me a thumbs up guys that everyone is actually clear about the same. Just focus on what we are learning right now, the names of the bonds. Yeah, it is basically C1 of the ribose sugar, okay. So I hope it's clear. Between N1 of pyrimidine, I'll just write it above, N1 of pyrimidine and C1 prime of ribose sugar. So that is the beta N glycosidic bond, okay, beta N glycosidic bond, okay. So now guys, now, yeah, now guys, coming on to the next, that is the nucleotide formation guys. That is the nucleotide formation. Nucleotide formation. Coming on to the next, that is the nucleotide formation. And remember, now suppose this is a nitrogenous base. This is a nitrogenous base guys, you already know this. This is the ninth place the nitrogen is there, and this particular nitrogen is basically joined by the first carbon atom of the pentose ring, that is the C1 prime, C2 prime, C3 prime, C4 prime, and we have CH2OH, and this carbon is basically the C5 prime carbon. Now this nitrogen is basically joined शुगर एंड फॉस्फेट will be present but a nucleoside. हाँ ठीक है, अब न्यूक्लियोटाइड में हम तीनों चीजें पढ़ेंगे ना. न्यूक्लियोटाइड इज बेसिकली मेड अप ऑफ बेस भी होगा सर, नाइट्रोजनस बेस होगा, पेंटोज शुगर होगा और फॉस्फेट ग्रुप होगा. पेंटोज शुगर विल बी देयर एंड अल्टीमेटली वी विल हैव द फॉस्फेट ग्रुप. अल्टीमेटली वी विल हैव द फॉस्फेट ग्रुप. सो गाइस रिमेंबर द फैक्ट कि जो N9 है इट विल ज्वाइन टू द C1 carbon of the pentose sugar by a beta N glycosidic bond. By a beta N glycosidic bond. Now guys, nucleotide basically involves the phosphate group addition. Now we will add the phosphate group guys. We will try to add the phosphate group, okay? And remember that addition of phosphate group is basically at the 5th carbon atom. It is over here, the phosphate will actually add, and okay, so remember that is done via a bond that is called as the ester bond. That is called as the ester bond guys. So remember that addition addition of phosphate groups, addition of phosphate group by ester bond to the 5 prime carbon atom, okay, and सिर्फ एक ही phosphate जुड़ा हुआ है. So this particular structure where we have the nitrogenous base, pentose sugar and the phosphate group so that structure is basically called as the nucleotide, and it is more precisely called as the nucleoside monophosphate, or in simple terms I will call it as a nucleotide. In simple terms I will call it as a nucleotide. So I hope till here everything is clear. I have just talked about 2 bonds, beta and glycosidic bond, the ester bond, okay? So just give me a thumbs up that everything is clear as of now. And now I will tell you how di and tri is formed. Yes, Meet, Nahid, very good guys. Very good guys. Usma, Junaid, Pavan, very good. Laris, very good guys. You all are participating in the lecture. Hansi, very good. Shakir, very good guys. So yes guys. Abhishek, very good guys. Sagar, yeah. Heather, Danish, Jaipur, very good guys. Okay, Rasab, very good. Lakshi, Suraj, very good. Now guys, try to understand one more fact कि सर अभी तो एक ही फॉस्फेट था, अब मैंने एक और फॉस्फेट लगा दिया यहाँ पर और फिर एक और फॉस्फेट लगा दिया, जब सपोज दो फॉस्फेट लग गए तो अब जो स्ट्रक्चर बनेगा दैट विल बी ओके न्यूक्लियोसाइड डाइफॉस्फेट, न्यूक्लियोसाइड डाइफॉस्फेट ओके। और फिर अगर मैंने एक और फॉस्फेट लगा दिया, if suppose I put one more phosphate over here and that is actually the nucleoside triphosphate, nucleoside triphosphate, triphosphate. Now guys, try to remember that addition of further phosphate groups is basically via bond that is called as the acid anhydride bond. It is called as the acid anhydride bond. Acid anhydride bond. Remember कि जब पहली बार फॉस्फेट लगता है तब कौन सा बॉन्ड होता है? एस्टर बॉन्ड। जब दोबारा फॉस्फेट के साथ जोड़ा है दैट इज कॉल्ड एज द एसिड एनहाइड्राइड बॉन्ड, और फिर फॉस्फेट दोबारा लगेगा दैट इज ऑल्सो कॉल्ड एज द एसिड एनहाइड्राइड बॉन्ड। एंड रिमेंबर व्हाट इज द सिग्निफिकेंस ऑफ दिस बॉन्ड? whenever this bond is broken, energy is released. Whenever this bond is broken, energy is released. Energy is released. Now guys, can anyone tell me the name? If suppose I put NH2 group here, can anyone tell me the name of this complete structure? I am taking the base as something, okay? I have given you a hint. I have put put NH2 group, okay? So can anyone tell me the name of this complete structure? What it will be? Guys, don't leave the class. Try to answer the questions. It's very, very important, guys. या पूरा नाम बताना है। जुनैद, फुल नेम? Adenosine triphosphate, that is none other than your ATP. Very good, Lakshi, very good. Adenosine triphosphate, and remember that is none other than, yes, Neha, Laris, very good. Junaid, very good. It is the ATP, guys. Yes, Heather, it's the ATP. और अल्टीमेटली मैं तुम्हें यही तो बता रहा हूं कि जब ये फॉस्फेट ग्रुप टूटेगा, व्हेन एटीपी विल ब्रेक इट विल गिव एनर्जी। और एटीपी में भी कौन सा बॉन्ड टूट रहा है? ये एसिड एनहाइड्रेट वाला बॉन्ड टूटता है, तो इट्स एन एक्सोजीनस प्रोसेस गाइस, एंड अल्टीमेटली देयर विल बी द रिलीज ऑफ एनर्जी. So that is the concept that I wanted to tell you in the nucleotide formation. That is the concept that I wanted to tell you in the nucleotide formation. Okay. Yes, yes guys, that is the reason I told you. Yeah, yes guys. Usma, very good guys. Just don't try to skip the class. Now we are going to discuss a very important thing. Okay, we are going to discuss a very important thing, guys. Yeah, now coming on to a fact that is called as the dinucleotide formation. Dinucleotide formation. Now guys, remember, suppose you all know that we have something called as the nitrogenous base. Or nitrogenous base is basically attached to the pentose sugar, the C1 sugar. Okay, I'll just draw it nicely. Yeah, so you all know that it is the pentose sugar through which these particular structures are attached. The base is attached to the pentose sugar, and it is the C1 C prime carbon, C2 prime carbon, C3 prime carbon, C4 prime, and ultimately we have CH2OH, that is the C5 prime carbon. And remember that this particular structure, basically a phosphate is attached, okay, and over here, if suppose this is the hydroxyl group. Now can anyone tell me कि कौन सा शुगर है, deoxyribose है या ribose sugar है? In the chat box guys, right now, first tell me which sugar is this, deoxyribose or ribose? I'm waiting for the answers guys. Yes. यस जुनैद फिर गलती कर दी ना यार, राइबोस शुगर पे तो यार दोनों जगह पे प्रेजेंट होगा ना, C2 और C3 दोनों जगह पे प्रेजेंट होगा। मैंने जब बोला सिर्फ C3 पे प्रेजेंट है तो वो डीऑक्सी हो जाएगा ना। हाँ लारिस, बाकी सब राइट, बाकी सब राइट। देखो सिर्फ C3 पे प्रेजेंट है तो डीऑक्सी राइबोस, दोनों जगह पे प्रेजेंट है तो राइबोस। कंफ्यूज ही नहीं होना, सिंपल ट्रिक दी है कि दोनों जगह पे है, 2 और 3 पे तो राइबोस और C3 पे है तो सिर्फ डीऑक्सी राइबोस। या so that is like okay guys, now guys try to remember कि ये एक न्यूक्लियोटाइड है, this is the one nucleotide, this is the first nucleotide that we have written। और अब मैं दूसरा न्यूक्लियोटाइड बना रहा हूं। अब हम डाई न्यूक्लियोटाइड की बात करें, डाई का मतलब दो न्यूक्लियोटाइड्स है, एक न्यूक्लियोटाइड तो हो गया बेस, शुगर और फॉस्फेट। अब मैं दूसरा न्यूक्लियोटाइड बनाने की कोशिश कर रहा हूं एंड जस्ट ट्राई टू सी इफ सपोज दिस इज द नाइट्रोजनस बेस, ओके, इट इज अटैच्ड टू द पेंटोज शुगर at the C1 prime carbon, okay, we have C2 prime or C3 prime and ultimately we have the OH group here, the CH2OH part over here, and we have the phosphate group here, we have the phosphate group here. So that is the second nucleotide, that is the second nucleotide guys, that is the second nucleotide. Now guys remember, a dinucleotide is formed when the C3 prime carbon, okay, now before coming on to the major aspects, remember guys कि free functional group पहले न्यूक्लियोटाइड में कौन कौन सा है, कि सर एक तो फॉस्फेट है और एक हाइड्रोक्सिल ग्रुप है। दूसरे न्यूक्लियोटाइड में भी it is the phosphate and the hydroxyl group, so we have two functional groups in both the nucleotides, whether it is the first one or whether it is the second one. Now remember that any nucleotide, whether it is first or second, remember the third carbon atom will actually attach to the phosphate group, okay, it will attach to the phosphate group, okay, and that is why, so it will be like, more precisely it will be like this. So I just have to show the phosphodiester bond, and remember that this phosphate PO4, I'll just write it like that, PO, okay, so that is the PO4, you guys can everyone appreciate, yeah. So ultimately we have the phosphoester bond over here between carbon and the phosphate, oxygen and carbon, it is the ester bond, okay, and phosphate over here it is also oxygen, over here it is the carbon, oxygen and phosphate, so one ester bond is over here also, so that combinedly this bond is called as the phosphodiester bond, phosphodiester bond, कि सर एक कार्बन, ऑक्सीजन और फॉस्फेट के बीच में एस्टर बॉन्ड, और एक कार्बन C3 वाला ऑक्सीजन और फॉस्फेट के बीच में एस्टर बॉन्ड, दो एस्टर बॉन्ड्स आपस में जुड़ रहे हैं तो फॉस्फेट है, डाई एस्टर बॉन्ड, एक ही फॉस्फेट है और दो एस्टर ग्रुप्स है तो डाई एस्टर बॉन्ड। एंड रिमेंबर इट इज फ्रॉम द C3 टू द फिफ्थ कार्बन, ये C5 प्राइम है ना, सो इट इज बेसिकली 3 to 5 phosphodiester bond, it is 3 to 5 phosphodiester bond, it is 3 to 5 phosphodiester bond. So the two nucleotides, okay, Everyone understood this concept, so we'll make some certain notes over here now. We'll make some notes over here if everyone has understood the concept कि हाँ phosphodiester bond समझ में आया सर. Apart from that, can anyone tell me the name of this particular bond कि ये A कौन सा bond है, ठीक है, और ये जो phosphate और CH2OH के बीच में कौन सा bond होगा, okay? A and B. Can anyone tell me? These are two questions for you guys. If you have listened to my lecture very carefully, you will be able to answer these two questions very quickly and smartly now. अमजद बच्चे देखो, ये CH2OH है, ठीक है, so that is the pentose part, okay. देखो ये जो है ये फॉस्फेट से अटैच्ड होता है PO4 से. अब ये PO4 को मैं इस तरीके से लिख सकता हूं ना, इस PO4 को आई विल जस्ट राइट इट द वे लाइक दिस, ओके. फॉस्फेट है, ऑक्सीजन है और ये है, तो अल्टीमेटली दिस इज अटैच टू द अदर कार्बन दैट इज ओवर हियर. यस, आंसर्स आर राइट. या बीटा एंड ग्लाइकोसिटिक बॉन्ड, या यही गलती. डाइएस्टर तो यार ये पूरा है ना, वो तो एस्टर बॉन्ड ही है, फॉस्फोएस्टर बॉन्ड. यस चुनेत रसब, डाइएस्टर तो ये पूरा है, और ये फॉस्फेट जो है इसके ऑक्सीजन के साथ, ओके, दैट इज द कार्बन, ओके, और यहाँ पे भी कार्बन है C3 प्राइम, C3 कार्बन ऑक्सीजन फॉस्फेट, तो ये एस्टर बॉन्ड एक हो गया, फॉस्फोएस्टर बॉन्ड, फॉस्फेट ऑक्सीजन कार्बन, कार्बन ऑक्सीजन फॉस्फेट, ये एस्टर बॉन्ड एक हो गया, तो दो एस्टर बॉन्ड हो गए, तो फॉस्फो डाई एस्टर बॉन्ड, फॉस्फो डाई एस्टर बॉन्ड. अमजद समझ में आया आप कि एक एस्टर बॉन्ड तो सर ये हो गया, एक एस्टर बॉन्ड ये हो गया, तो ये फॉस्फोडाइएस्टर बॉन्ड है, और ये 3 प्राइम से लेके ये कौन सा कार्बन है C5 प्राइम, तो ये 3 से 5 तक रन करता है. इट इज रनिंग फ्रॉम 3 to 5. इट इज रनिंग फ्रॉम 3 to 5. Is it clear guys now? Yeah, everyone is right. Everyone is right. Phosphoester bond. Yes लक्ष्य, एक ही फॉस्फेट के बीच में है ना, CH2OH से फॉस्फेट जुड़ा हुआ है, तो फॉस्फेट है, ऑक्सीजन है, कार्बन है, फॉस्फेट ऑक्सीजन कार्बन. इट इज द फॉस्फोएस्टर बॉन्ड. इट इज द फॉस्फोएस्टर बॉन्ड, ठीक है. C3 प्राइम का कार्बन ऑक्सीजन है, फॉस्फेट है, तो ये एक एस्टर बॉन्ड, ये फॉस्फेट ऑक्सीजन और कार्बन ये एक एस्टर बॉन्ड, तो दो एस्टर हो गए, फॉस्फोडाइएस्टर बन गया. तो डाइन्यूक्लियोटाइड बनाने के लिए विच बॉन्ड इज नेसेसरी? इट इज द फॉस्फोडाइएस्टर बॉन्ड. फॉस्फोडाइएस्टर बॉन्ड. It is the phosphodiester bond guys. Okay, I hope everyone understood as of now. I hope everyone understood as of now. Yes, everyone understood as of now. Yes, crystal clear. Okay guys, that's what I wanted. Now guys, we'll try to write some points. Yeah, we'll try to mention some points now. Remember, dinucleotide formation, the two nucleotides are joined together by 3 prime to 5 prime phosphodiester bond, and it is between the 3 hydroxyl group of the first nucleotide, okay, to the 5 prime phosphate group to the other nucleotide. Apart from guys, remember, apart from this, okay, remember कि the nucleotide with a free functional group, it is at 5 prime position, okay, and that is referred to as the first nucleotide. and remember it is also called as the head end of the nucleotide. It is also called as the head end of the nucleotide. नाउ हाउ सर, बताइए फिगर से। अब ध्यान से देखो कि ये जो हमने पहला वाला जो न्यूक्लियोटाइड लिया था, इसका सर ये जो दिस वाला जो फॉस्फेट है, ओके, दैट इज एट C5 प्राइम कार्बन पे ये जो फॉस्फेट ग्रुप लगा हुआ है, ये फंक्शनल ग्रुप जो लगा हुआ है, दैट इज फ्री। ये तो फ्री है सर, ये तो बॉन्ड बनाने में कहीं पर भी यूज नहीं हुआ। सो न्यूक्लियोटाइड विद अ फ्री फंक्शनल ग्रुप एट 5th पोजीशन, इट इज ऑलवेज कॉल्ड एज, बाय कन्वेंशन हम लोग इसको फर्स्ट न्यूक्लियोटाइड लेते हैं, अदरवाइज हम इसको सेकेंड भी बोल सकते थे, ओके, एंड दैट इज कॉल्ड एज द हेड एंड ऑफ द न्यूक्लियोटाइड। दैट इज कॉल्ड एज द हेड एंड ऑफ द न्यूक्लियोटाइड। कि सर कि इस पर्टिकुलर न्यूक्लियोटाइड में दो फंक्शनल ग्रुप थे, एक C3 प्राइम कार्बन पे और एक C5 प्राइम कार्बन पे। तो C3 वाला तो यूज हो गया बॉन्ड में, C5 वाला बच गया। जो C5 वाला बच गया यानी कि फॉस्फेट ग्रुप जो यूज ही नहीं हुआ बॉन्ड बनाने में, सो दैट इज द फ्री फंक्शनल ग्रुप। और ये कौन से पोजीशन पे है? 5th पोजीशन पे है। सो दैट इज कॉल्ड एज द फर्स्ट न्यूक्लियोटाइड और कॉल्ड एज द हेड एंड। कमिंग ऑन टू द नेक्स्ट पार्ट, the next statement, the nucleotide with a free functional group at 3 prime position, it is called as the last nucleotide. It is called as the last nucleotide, and remember it is the tail end, and remember it is the tail end. The nucleotide with a free functional group at 3 prime position, it is the last nucleotide called as the tail end. Now guys, see this particular image, the second nucleotide। कि सर इसमें कैसे पता चलेगा? अब you can see कि सर थर्ड पोजीशन पर देखो, इस पूरे न्यूक्लियोटाइड में दो फंक्शनल ग्रुप है, एक ये वाला फॉस्फेट और एक ये वाला हाइड्रोक्सिल। सर C5 वाला तो बॉन्ड बनाने के लिए यूज हो गया, तो बचा ये C3 वाला। सो दैट इज द फ्री फंक्शनल ग्रुप, दैट इज द फ्री फंक्शनल ग्रुप। और जो C3 प्राइम वाला है, जो थर्ड पोजीशन पे फ्री फंक्शनल ग्रुप है, ये OH तो कहीं यूज ही नहीं हुआ। दिस OH इज नॉट यूज्ड इन द बॉन्ड फॉर्मेशन। सो दैट पर्टिकुलर न्यूक्लियोटाइड जहां पे C3 वाला जो है, which is not used in bond formation, it is called as the last nucleotide. It is called as the last nucleotide, and remember it is the tail end. Okay, it is the tail end of the nucleotide. It is the tail end of the nucleotide. इसलिए बार बार तुम लोग पता है बुक्स में पढ़ते हो 5 प्राइम टू 3 प्राइम डायरेक्शन, तो ये 5 प्राइम तुम अब ध्यान से देखो बच्चे, दैट इज द एक्सप्लेनेशन विच आई एम जस्ट टॉकिंग अबाउट कि 5 प्राइम तो सर ये फ्री फंक्शनल ग्रुप है 5th पोजीशन पे, जो पहले वाला न्यूक्लियोटाइड है, और 3 प्राइम हाइड्रोक्सिल ग्रुप है जो फ्री ग्रुप है सेकेंड न्यूक्लियोटाइड में। सो मेजॉरिटी ऑफ द टाइम्स इन होल ऑफ द बुक्स, इन होल ऑफ द नोट्स यू विल फाइंड इट कि हाँ सर 5 प्राइम टू 3 प्राइम डायरेक्शन में रन करते हैं, ठीक है। सो दैट इज द रीजन व्हाई यू गाइस हैव टू बी वेरी मच पर्टिकुलर। Apart from that guys, if you want ki remember that basically they exhibit polarity, so we'll write one more point over here that these functional groups are ionizable, okay. Basically they exhibit polarity. You all know that they are dissolved in water and they are the ones which will actually exhibit the polarity, okay. And by convention, remember the base sequence of nucleic acids, it is written from 5 prime to 3 prime. It is written from 5 prime to 3 prime. So you can easily see that we have the phosphate, okay, we have the sugar, and we have the base. Nitrogenous base phosphate sugar. It is nucleotide 1. Now this sugar is basically attached with a phosphate to the another sugar, okay, and the base is there. Again the same thing. The phosphate will be there, the sugar will be there, and it will be attached. Now this sugar will always have a free functional group OH at 3' end, and this particular sugar will always have a phosphate group at C5 end. So it is always by convention 5' से 3' end लिखते हैं, and that is the free functional group that is present. That is the free functional group, guys, that is always present. That is the free functional group that is always present. So yeah, by convention it is like that, and yeah. Yes. So just give me a thumbs up that, guys, you all understood this very well, and we'll just recap all the things now. Just give me a thumbs up that all of you understood it. Yes. Junaid, okay. What about rest, everyone? Yes. Hansi, Kapil, Usma, Manish, Laris, Amjad, Abbas, Pavan, very good. Very good, guys. Shakir, Heather, very good. Lakshi, very good. Very good, guys. I'm totally impressed that you all guys are listening me out. Yeah. Very good. Very good. Sakit, very good, guys. Well, you won't regret after this lecture because everything is taught in so much detail now. Basics should be clear, and yeah, that too, yeah, it's taught in so much detail. Ronak, very good. Dharmender, very good, guys. So we'll just summarize this with the help of a table right now. You all know that we are discussing about the ribonucleotide, and we can also have the deoxyribonucleotide just based on the sugars. Pentose sugar, whether it is deoxyribose or ribose. So drawing a table right now, you should know we have the different nitrogenous base. Yeah, Shabreen, Shrimali, Dharmender. Okay, Jaipur, very good. The nitrogenous base, the nucleoside. What will be the ribose component? Ribose nucleoside monophosphate. And we have the deoxyribose nucleoside monophosphate. आज आपकी वजह से बेसिक्स क्लियर हो गया सर. Thank you, Junaid. If I'm somewhat helping you guys in any way out, it's like for me it's also a very good thing. Okay, so in any way out, if I'm like helpful to you guys, I think it's a compliment to me as well. Apart from that, guys, if suppose I take the nitrogenous base as adenine, so what will be the nucleoside, guys? When the adenine, nucleoside का मतलब क्या होता है? Base, nitrogenous base plus sugar. So it will be adenosine. Adenosine. It will be adenosine monophosphate. It will be adenosine monophosphate. And if it is deoxyribose nucleoside monophosphate, so it will be deoxyadenosine monophosphate. Now, guys, coming on to the next one, that is the guanine. And remember that guanine, the nucleoside, it will be guanosine. It will be guanosine monophosphate, and it will be deoxyguanosine monophosphate. guanosine monophosphate. Monophosphate. Coming on to the third, that is the uracil. Uracil. You can easily appreciate uracil is basically the uridine. It is called as the uridine in its nucleoside form. Okay, it is the uridine in the nucleoside form. Okay, so you can easily write it down as the uridine monophosphate, and you can mention it as deoxy. Okay, okay. Now guys, tell me, we'll have this component or not? So apart from that guys, you will also have to tell me about hypoxanthine. Hypoxanthine, xanthine, cytosine. Xanthine, cytosine. So I will just tell you the nucleoside name, that is the inosine, xanthosine, and cytidine. Okay, so you guys will be the one who will complete this table and will text on to my WhatsApp giving me all the possible names. Now I've just told you the nucleoside name, the inosine for hypoxanthine, xanthine का xanthosine होगा, and the cytosine, it will be cytidine. So you guys will be the one who will actually tell me this only particular names in my particular in my particular. यार ये बिल्कुल होगा, it will be definitely, it will be there. Yes, definitely it will be there. It will be not. No, I was just checking it. यार वो तो शुगर की बात कर रहे हैं ना यार. शुगर तो कोई भी हो सकता है, राइबोस शुगर भी हो सकता है, डिऑक्सी राइबोस शुगर भी हो सकता है. Okay, so uracil, and it's not the DNA or the RNA. Okay, it's not the DNA or the RNA. It will be definitely there guys. Okay, it's not the DNA or RNA, and it will be actually deoxyuridine monophosphate. Deoxyuridine monophosphate. Understand the lecture at once more guys. एक बार और देख लेना कि हम यहाँ पे शुगर की बात कर रहे हैं. शुगर कैन बी राइबोस शुगर ऑल्सो और डीऑक्सी राइबोस शुगर ऑल्सो. ओके, तो यस देखो आरएनए भी तो डबल स्टैंडर्ड्स होते हैं ना. आरएनएस आर ऑल्सो डबल स्टैंडर्ड ना. So yeah, no, you have to be very much perfect क्योंकि हम राइबोस शुगर की बात कर रहे हैं यहाँ पे. Okay, so guys, just give me a thumbs up that you all understood today's lecture very well, and please don't forget to watch this particular video. Don't forget to watch this particular video later. Also you can share your doubts with me on my WhatsApp. You can join the Telegram group for further more questions. Yes, Abbas, Lakshi, Heather, Junaid, I just want to see that everyone is over here or not, and you can join my further Telegram group for that. Yeah, Ronak, Kasim, Dharmendra, Laris, Usma, Amshad, Rasab, very good guys. I have tried my level best for you guys to explain you all the concepts with I will definitely share this PPT on my Telegram channel. Okay, so it's better if you join that, and we'll definitely work. Okay, we'll definitely work over there also. Okay, not an issue as well guys. So just share your feedback guys. Definitely. So after some time I will be removing the video. If anyone wants, ask him to watch the video, because it was unnecessary that everyone should join the class whenever it is scheduled. Okay, watch the video once again. You will get the crisp, concise way of all the basic nucleotides. you'll get the grips. Your teaching level is ultimate. Yeah, pro कहते हैं ना उसे, the that one pro is there. Yeah. वेलकम लक्ष्य, वेलकम अभिषेक, अमजद। ये 25 तक कवर हो जाएगा। मुझे अगर 3 महीने 4 महीने देते तो मैं कह देता हाँ। नहीं देखो, मॉलिक्यूलर बायोलॉजी में मॉलिक्यूलर बायोलॉजी टेक्निक्स भी आते हैं, बहुत कुछ है मॉलिक्यूलर बायोलॉजी टेक्निक से आजकल नीट पीजी में क्वेश्चंस काफ़ी आ रहे हैं। सो अल्टीमेटली इट इज़ द बेसिक फंडा यू गाइस हैव टू अंडरस्टैंड कि हाँ कि क्या इम्पोर्टेंट है, ये सब चीज़ें जानना बहुत ज़रूरी है। आगे व्हेन आई विल डिस्कस द मेटाबॉलिज्म ऑफ प्यूरिन्स पिरामिडिन्स, तो स्ट्रक्चर्स जानने बहुत ज़रूरी हैं। That's why in the very first lecture I discussed the structures, because that was ultimately required for the metabolism also. और मेटाबॉलिज्म, सैल्वेज पाथवे सब तब समझ में आएगा जब स्ट्रक्चर्स आते होंगे, तब मतलब बिल्कुल क्लियरली तुम लोगों को समझ में आ जाएगा। सर ये 25 तक कवर हो जाएगा। नो अमजद, आई कैन नॉट गारंटी यू एनीथिंग, बट हाँ रेप्लिकेशन से क्वेश्चंस बहुत ज़्यादा आते हैं। सो देखो, सिर्फ एक पर्टिकुलर एग्जाम के लिए नहीं पढ़ो। आई ऑलवेज से दैट प्रिपेयर योर नोट्स फॉर द लास्ट, आई मीन फॉर योर नेक्स्ट एग्जाम ऑल्सो। इसमें इतने क्वेश्चंस हैं जो पहले भी एग्जाम में आ चुके हैं, जो नोट्स हमने लिखे हैं। सो दैट इज यस, कमलेश डीऑक्सीराइबोस शुगर था। हाँ, anatomy I will continue with the scapula later on. Yeah, I'm always there for you guys. If you are respecting me, if you are following what I'm saying, if you are attending my classes without fail, without excuses, and if you are maintaining the notes, I mean that's the way I'm quite impressed, and I'll be helping you out if you are actually following me. Okay, that's the thing that is required. Yes, 8 number. खान साहब थे क्लास में चलो, very good guys. Okay guys, that's all for the session today. I'll just quit the class, and if anyone wants, he can join the channel. The number is already displayed on the screen, and we will Rahul Singh. Yes, okay guys, I'll just end the class now. Goodbye, always with us. Heather. Okay guys, bye. नोट्स बना रहे हो, वेरी गुड यश, वेरी गुड डे अंशे, वेरी गुड। चलो एनीवेज, सो आई जस्ट एंड द क्लास। थैंक यू अंजलि। ओके अंजलि, ओके एंड द